On Lab Matters today with me, Harriet Gould, we've got award-winning and groundbreaking synthetic biologist and poet, Professor Anne Osbourn. Join us to hear Anne's views on specialising in your subject. Today we have with us Professor Anne Osbourn, OBE, FRS and NAS as well. We'll ask about those, what that means later. Anne is one of the brilliant group leaders at the John Innes Centre, which is the Centre for Excellence in Plant and Microbial Science. So that gives you a clue of what she's up to. And Anne, through your research, you've made it possible to combine genomics and synthetic biology in new ways, which will be useful application-wise in agriculture, medicine, and in industry. So we'll look forward to hearing about that as soon as possible. But first, I thought it might be nice to get to know you a bit before we go on to your work. And so perhaps you can tell us a little bit about how you grew up, where you grew up, and what that was like.
Yeah, so I grew up in what at the time was a little village in Yorkshire called Eldwick, which was on the non-posh side of Ilkley Moor, really.
The non-posh side.
Near Bingley. So my parents were from Lancashire and they migrated to Yorkshire before I was born, but that was a big move and they were still not quite accepted.
Wow.
So it was a lovely little village. And I remember going for walks with my mother and I was very interested in plants. We would go out, I'd make her go out in the snow to find the first coltsfoot flowering by the stream. And we go for picnics and she put up with a lot really. But apparently when I was 3, I could say primula denticulata. So I was probably quite precocious.
I
I
wonder
just
where you
love
learned that.
flowers. Yeah. Well, she must have told me, I suppose, but I just love flowers and I remembered gardens more than people's houses. So I navigated by flowers and that took me into working on them eventually, onto plants at least.
Yeah. And was that could was that supported at school when you were at school, or did anyone try and think don't focus so much on plants, or was it very much encouraged?
Well, I suppose at primary school, I can't really remember primary school, but it was, you know, I just remember the field with the buttercups and the daisies on it. That's what I remember from the outside of the school. Where Fred Hoyle he went to school at the same primary school as me, Eldwick Primary School.
Wow.
So he would have sat on that field contemplating the universe when he was about 5, probably.
No doubt.
And then I went to grammar school, Bingley Grammar School, where I enjoyed creative writing a lot. But then when it came to A levels, to specialisation, the curriculum wouldn't allow me to do English as well as the sciences. And I decided I wanted to do biology. And so I ended up doing the biology, chemistry and physics. And I had to leave English behind, which I was very sad about. So then obviously I was focusing on science and we did quite a bit of plant science in biology. But that was when I started to become a specialist.
Mm. And then I suppose it was lucky in a way that you did do all three, because all three probably quite important in what you've been doing. I don't know if it would have been different if you had maybe, I don't know, not done chemistry, for example. And
Yeah, I think chemistry was very important to me. Physics, I thought I was good at, but actually I wasn't. And that's why I didn't get into Oxford in the end.
wow.
So I'm what you might call an oxymoron.
Remember
Um,
that one?
so no, I mean, when I was doing my A levels, the girls were being encouraged to go down the scientific route, which I suppose was good. And also my parents were both literary specialists. So I was from a background of English literature. And my parents' house was full of his and hers copies of all the classics double stacked on bookshelves. And so my sister and I think, both thought, well, that's there anyway. So we can always go back to that and talk to them about literature. So we both became scientists.
Mm-hmm. I wonder, it's interesting. This comes up so many times, in fact, that that differentiation between the sciences and the technical subjects and the arts. And would it be better if the curriculum did have more sympathy towards the two of them being learnt together?
I think in this country we tend to specialise quite young.
Mm.
So even when children leave primary school and go to high school, they start to narrow and become quite, the disciplines become very partitioned.
Mm.
And it's important to have partitioning of the disciplines so that they are taught to a high standard and they are discrete, but also it's important to be able to think across them.
Mm.
So there's a fine balance there between having high quality teaching that does focus on disciplines and having the flexibility to think more widely. I think the key thing, though, is for children to develop ownership of learning.
How do you think that that can be done more effectively?
So one of the things that I accidentally stumbled across a number of years ago, because of my interest in poetry, so I'm a poet, I am interested in poetry, was the idea of using really stunning scientific images to try to, initially just I was trying to explore science in creative ways, starting with images as one form of prompt. So remarkable, surprising images, not obvious ones, like a thermogram of a cup of tea being poured, for example, something under the microscope or something in the universe. And it occurred to me that those kinds of images intrigue people, whatever their backgrounds are, whatever their age, they intrigue people and draw them in.
Mm.
So while I was exploring this myself, I also went into the local school, the village where I live now in Norfolk, where my younger daughter was still a primary school student, and spoke to the teachers. And we set up a project by accident, really, that involved using images to draw children into interest in practical aspects of science, for example, microscopy. And then it grew into a project where after they'd done the science, they did art and they did creative writing, but all exploring the same sorts of themes. And it just transpired that this kind of approach meant there was a way in for children with different interests, different learning styles. And because it was all focused around a particular theme, which could be outer space or under the microscope or whatever else, it became a way of exploring in different ways, not just through practical science, but through artistic exploration as well, which gave a deeper understanding and interest in the overall theme.
Mm. Yes, I can see how that would work. Because I suppose if you tell somebody, oh, this is something you ought to be interested in, that feels a little bit prescriptive and almost that it might second guess someone's own preferences. Whereas if they it's naturally drawn out of them, as I suppose education means it, then that's it. They're on the path to owning their own learning.
It has, I mean, we've run a lot of these projects. Now, this started out as a one-off project, and 20 years on, it's now a big initiative. And
Mm.
we work with hundreds of schools in the
That's
UK, in China, elsewhere. So it's called the Science, Art and Writing Project, www.sawtrust.org.
I
It's a
see.
charity.
I saw it in your bio and I didn't realize that that was what it I didn't realize that that's what it was about. I thought it was about simply about I mean, I don't know what I thought. I just thought it was more related to your poetry, I suppose. But yeah, that's a really holistic approach. Interesting. Um okay, so look, on to back to you really and your journey through to being a scientist. So after A levels, you went to
I went to Durham University to study botany
okay.
because of my long love of flowers and plants, and also because I was squeamish. And so dissecting animals wasn't something for me. I outsourced that to my friend Susan in biology at A level. And so I couldn't have been a doctor or a vet. I would have just fainted. So I went into botany. And
Was it,
Durham had a really good broad-based course. And when I first went there, David Bellamy was still there as well.
gosh, treat. What a treat. Did
Mm.
you and you worked with him?
Yeah, yeah.
Hmm. Do you remember any of the projects?
Well, I was in his tutorial for a while and we did all sorts of things, including taking roles in protein synthesis and simulating this, and I was slow to speak up at the time, so I got cell organellogenesis as my role, which wasn't good. We also went up to the Scottish borders, and we were by a lake, or a lough as it was, a peat lake up by Hadrian's Wall, and I think it was February. And I remember asking David Bellamy what that plant was that was growing in the water. So he waded in, picked some of it and brought it back. Tragically, I've forgotten what it was, but it was nearly snowing.
Yeah. And he'd gone into the bog
Yeah.
to retrieve it. Yeah, I suppose when you're that hands-on and you're surrounded by people that immersed, it can be a fairly inspiring experience.
Yeah. Yeah,
Mm.
absolutely.
Mm. Um, and then you graduated. I mean, I've looked on your LinkedIn. Everything you've done has been extremely high, high achieving, honors and firsts and all sorts all over the place. You know you're. I don't think there's maybe anyone better than you in some respects, but
That's true.
um, but so when you graduated, did you have a clear idea at that point what your next step was going to be?
So when I was doing my degree, so I was an Oxford reject, and I went to Durham, where there's still a college system. And I remember going there from my grammar school, thinking that I'd always been top of everything at school. And then I got to university, and we had exams at the first Christmas in December, and I was 42nd out of 84. And I remember thinking, okay, so I was quite pragmatic about it. That means, in the big wide world, that's where I am. I'm average. But then by the end of it, I was one of two who got first class degrees.
Mm.
So I think a lot of it was normalizing out people from different backgrounds where the teaching had been perhaps a bit more university oriented. But I'd basically we had very good teachers at school, but I'd very much been brought up to just do things myself.
Mm-hmm.
And so during my degree, I remember in the third year, one of the lecturers said, you could actually do a PhD if you wanted to. And I thought, really? I always thought I was stupid. But then I thought, well, maybe. So I started looking for opportunities and applied for several different PhDs. And in the end, I got one at the University of Birmingham in the Department of Genetics. So I went there to do a PhD, working on understanding at the mechanistic basis the interaction between wheat, barley, and a particular fungal pathogen that causes diseases of cereals called gloom blotch disease. And that involved learning genetics and learning how to work with fungi and doing disease tests and so on.
Mm-hmm. And from that, well, I mean, you obviously enjoyed that enough to continue, and it's like you say, it's been a theme since you were 3 years old. So then you got your PhD. I mean, did you remember your tutors from that time?
Oh, yes. Yeah. My PhD supervisor was Chris Caton, who is a lovely man. He's retired now, but I learned a huge amount from Chris, and he was so patient and very, very smart. So he was always there if I had any questions. He challenged me. And so he's been very, very much instrumental in shaping who I am.
Wonderful to have that sort of influence around. So then what after that was your first job?
Well, towards the end of my PhD, I decided I still very much like research. I liked, in a sense, having ownership of my learning, identifying problems, devising ways of addressing those problems. So I was really enjoying scientific research. And at that time, which was the mid-1980s, a long time ago now, molecular biology was a big thing. So people learned how to clone genes and sequence DNA and this kind of thing. And those methods were beginning to be applied to studies of plant diseases and understanding more at that sort of level. And it just so happened that somebody in Norwich, Mike Daniels, had started using these approaches to understand how a particular bacterial disease infects turnips, it happened, brassicas. And so I had an opportunity to come and work with him to learn the sort of the cutting edge methods, how to work with DNA. So this is a long time ago now, and young people will think, my goodness, she's that old. It was before the fax machine and definitely before the internet. So I cut my teeth on that sort of work during a 2-year postdoctoral position here in Norwich, where I am now. And then I've actually stayed in Norwich ever since then, but in various guises. I haven't been static. I've had different roles and moved around, just haven't been far geographically.
Mm. And I want to talk about your real specifics about the research that what you're doing now, um, your discovery, in fact. I don't know how many people get to have a discovery to their name, but you certainly have. Can you talk us through that?
Well, I suppose over the years, that means my research group have made a lot of discoveries. And initially we were working on this understanding how plants and diseases interact. But that led me into wanting to know more about the chemicals that plants make, because plants make all kinds of chemicals, the scents, the colors, the flavors that we associate with different types of plant. And they don't just do it for fun. They do it because these molecules are serving a role. They might be attracting pollinators or repelling insects, or they have roles in ecological interactions. So I became interested in the roles of plant chemicals in protection against diseases. And that's what took me into moving away a little bit from working on plant disease interactions ultimately, because I then became fascinated with how plants make chemistry, because the plant kingdom collectively is capable of making an enormous array of different kinds of chemicals, most of which we haven't yet discovered. I mean, the estimate at the minute is 1 million, but it'll be a lot more than that. Mm-hmm. And these chemicals have the potential to be the next generation of drug leads and other useful molecules. And one of the things that my group found quite a few years ago now was that, very surprisingly, so these chemicals are complex molecules, and to make these chemicals, plants need a number of genes encoding enzymes that catalyze the different steps in the pathway to make a complex molecule. So that might be 10 genes. Like we have 1 gene that determines eye color, 10 genes to make a complex molecule. And people thought that these genes for these different natural product pathways in plants were scattered around plant genomes. And that's certainly true for some of the molecules that we're very familiar with, like the flavonoids and the colors, the pigments in plants. But we found an example where, in fact, the genes for one of these multi-step pathways were right next to each other in the genome, like beads on a string. And that was a big surprise at the time. There had been one other report from a group in Germany for a different kind of molecule made by maize. We were working with oat. And it turns out that actually these biosynthetic gene clusters, as they're known, are quite common in the plant kingdom. And there are lots of intriguing questions around how these gene clusters arose. But also it's very practical, because if you have the sequence of a plant genome and you have a computational algorithm you can use to scan the genome to look for these beads on a string type things. So you don't necessarily know what these things are doing, but you can predict that if you see a string of genes that look guilty, they look like they might be encoding enzymes that make chemistry, then it's a very quick way of ripping through a plant genome sequence and finding potentially useful genes. So that's kind of revolutionized the way that we think about how to harness the chemical engineering capability of the plant kingdom. So that's been a big advance that's raised a lot of interest.
And the application areas are quite varied. And do you have anything to do with how it's being used in practical terms now?
Yes, because these kinds of molecules, that the natural products of the plant world, as I say, they have all kinds of functions. I mean, we're working on a particular compound that is it's an insect deterrent. It's produced by the neem tree, which grows in Asia, and the relatives of the neem tree. And there's a lot of interest in this molecule because it's friendly to bees, for example. So it's regarded as an environmentally sustainable alternative to crop protection. If we can learn how these chemicals are made, then that could help to devise the next generation of environmentally friendly insect deterrents and insecticides. But most of our efforts are on molecules that have potential drug applications. And I've recently, with others, spun out a company called Hothouse Therapeutics based on this technology.
Say that again,
Hothouse Therapeutics. It doesn't yet have a website, but it will have soon. It started in July, so we've got about 2.5 million of seed investment, looking to raise a bit more. But it's off the ground now, so that's exciting.
And when do you think that'll be live?
The website?
Yeah.
Fairly soon. We're sort of in stealth mode at the minute, really, because we want to get some stuff done before we make a big ta-ra thing. So when the website does go live, it'll be fairly low key.
Understood. So just going back to that, the as you say, environmentally sustainable way of doing insect repellent. I suppose that'll have a knock-on effect, a positive knock-on effect to water quality, because it'll reduce the pollutants going into groundwater and so on.
Yeah, absolutely.
Which I suppose will in itself increase the quality of the soil, and that perhaps isn't the downside.
Yes, I mean, we obviously, we're all well aware now that we have to do what we can to protect the planet and to protect biodiversity. We need to move away from using fossil fuels to make chemicals, for example. If we can harness the enzymes and the chemical engineering capability of the plant kingdom and other organisms to make things, then we can do that using biosynthesis rather than chemical synthesis. And we had a project that we're still working on, but it's a nice story about a tree that grows in a certain part of Chile, in the middle of Chile, and it's called the soapbark tree. And it's called the soapbark tree because for hundreds of years, the extract of the bark has been used as a natural soap or shampoo by the indigenous people of Chile. Excuse me. And but importantly, it turned out in the early 1900s that this bark extract also has another very important property, which is that if you put it into a vaccine, and this work was originally done by a French vet, if you put a bit of bark extract into a vaccine, you get a much bigger immune response. And so there's something in this bark extract that is an immunostimulant, also known as a vaccine adjuvant. And quite a few years on, the active compound was purified, and it has a catchy name of QS21 because it was fraction 21. And QS21 was approved for use in human vaccines in 2017. So it's now in vaccines such as the shingles vaccine, Shingrix, malaria vaccine, COVID vaccine, and around 40 or so other vaccines currently under development. So this molecule is hugely valuable. It's currently worth between $100,000 and $400,000 a gram. And it's still being sourced by extraction from the bark of wild trees in Chile. And the trees need to be 20 to 25 years old to be big enough to make enough of the molecule for that to be worth doing. So there's concern about the environmental sustainability of the supply chain for this molecule. And so we sequenced the genome of the tree, and we've worked out how the tree makes the molecule. And we've put the steps needed to make the molecule together and put them into a different plant, a big leafy green plant that we grow routinely in the glasshouse, which is called Nicotiana benthamiana, and it's a wild relative of tobacco. And so we can now make this molecule free from tree, as they say.
Mm.
So that's really exciting because it opens up opportunities to then not only make QS21, but to make different flavors of QS21 for use as vaccine adjuvants.
Mm-hmm. And I wonder, are there any risks involved in that type of biosynthesis in terms of potentially diluting the potency of nature itself? Or is it in that completely controlled environment going to be consistent forevermore?
When you say diluting the potency of nature, can you
Yeah. So, I mean, for example, just trying to synthesize something that copy what's happening in nature, would it ever become less than it would be in its natural environment?
Well,
Or
I think that by being able to do this, it means that the tree, I mean, there's a lot of optimization to do, but ultimately it should be possible to produce this molecule, hopefully at scale,
Mhm.
without taking the bark off the tree, or at least as a complementary means of supplying the molecule, because the tree is, you know, there are only a limited number of trees and the bark is being stripped off them, and this is not good for the tree. So having spoken to the Chilean botanist Gloria Montenegro about our work a while ago, she was very excited about the fact that what we're doing is helping the Chilean forests to survive.
Oh, good. So there's because I mean as well, because I suppose what they benefit from a financial perspective, but yes, there is a risk to the biodiversity in that area of the world. So it's
Yeah, as I say, the demand for this molecule is growing massively, so it will soon outstrip the
the forests.
forest. So it is a sensitive matter, but you know, at the moment, I think there's generally a very upbeat feeling about the fact that there's a backup plan.
Yeah, definitely. Um, so that's your science work, and let's move quickly on to your creative outlet, which you've mentioned a little bit already, but you are in fact a poet. And I suppose this was, yes, influenced by your parents being from the literary world. But but doesn't being a poet, you know, something has to come out of you. You need to have something to say. How did you first realize that was what was happening?
So I always loved creative writing at school. I read poetry at school. I didn't write poetry for the most part, but I love creative writing. And then, as I said, I had to put that on the back burner because I was doing science. And then I spent years writing scientific papers and reviews, which are all very, what's the word?
Slightly
Clinical,
drier. Yeah.
dry, and everything has to be referenced. And so having done this for a very long time, I was beginning to think, actually, you know, I am established as a scientist. I've published lots of papers, but things didn't seem to be quite right. And then I saw an advert in Nature magazine in the jobs section for the National Endowment for Science, Technology and the Arts, NESTA program, which no longer runs, unfortunately, but it was called the Dreamtime Fellowship Program, where if you were established in science, technology or the arts, you could apply for one of these fellowships to spend up to a year doing something radically different. And the idea was that you could take the benefits of that experience back to your day job. So being in Norwich, the John Innes Centre where I am is 10 minutes walk from the University of East Anglia, which is where the School of Literature and Creative Writing is. And so I decided, rather radically, that I wanted to apply to go to the School of Literature and Creative Writing for a year to explore my creativity in other ways, which was a challenge because firstly, I didn't know anyone there. And secondly, I didn't have English A level. But through a friend of a friend, I ended up being put in touch with somebody who'd done their PhD there, who introduced me to her supervisor, Clive Scott, who at the time was head of the school. And so Clive was very open to radical things. And we had a coffee, and he was very excited about the idea of hosting me in the School of Literature and Creative Writing. So I wrote a Dreamtime Fellowship proposal, which was pretty woolly, to be honest. But the overall thrust of it was that I wanted to go and explore my creativity in other ways, with a few things in there that made it sound credible. And I got an interview. I went down to London for an interview. And there was a neuroscientist there, and then two people from Nesta. And the senior person from Nesta, I think, had a theatre background. And I gave a 10-minute presentation, and they asked me questions. And then I was asked if I had any questions for them. And the lead Nesta person said, yes, what are your views on tangible outcomes? So, no, sorry, I said to her, what are your views on tangible outcomes? And she just went, sometimes it happens, sometimes it doesn't. And I thought, yes. Anyway, luckily, I got one of these fellowships. But then I didn't know how to tell the people at work that I was actually going to go and write for a year. So I deferred taking it for as long as possible, so I didn't have to tell people until I'd worked out how to do it. But in the end, I managed to set it up so I could keep my group running, come in one day a week, and the rest of the time I was out in the wilderness writing. And that's when I accidentally started writing poetry, which was most disturbing, because I had no idea that I would be doing that. And then I spent a year. I mean, the fellow writers that I met were so kind and patient. I mean, because my poetry was awful.
Were they from different, presumably they were from all sorts of different backgrounds as well, not necessarily literature.
So the Nesta Fellows were from, yes, very diverse backgrounds, but I was I was a visiting fellow in the School of Literature and Creative Writing at UEA without an English A-level. So I did a fellows reading next to John Boyne, who was writing The Boy in the Striped Pajamas, in front of Rose Tremaine and Richard Holmes and all of these famous people.
Mm.
And it wasn't a taught course. I had 1 hour a week with Clive, my mentor. The rest of the time I was just, I say in the wilderness, but at that time Norwich had set up, at the time it was called the New Writers Centre, or New Writing Centre. It's now the National Centre for Literature. I'm getting this wrong. But luckily there was a workshop that I went to where I met a lot of poets who were very, very long-suffering and kind, as I say. And that meant that I had people I could send my offerings to and people I could talk to, and gradually get to learn more about the world of poetry, which I'm still grappling with.
And it's interesting because, of course, poetry is written down, and one never really knows the true interpretation, perhaps, behind it, unless you're lucky enough to meet the person who wrote it and they can explain it, or perhaps even read it in the way that it was supposed to be read. How do you feel about in your interpretations of other people's poetry and how you think that your poetry is going to be interpreted?
Well, I suppose as a scientist, as scientists are not used to sharing their emotions or feelings. That's one thing. But of course, a poem doesn't have to be an autobiographical poem. That's up for debate. I used to go to the literary festival events at the University of East Anglia, where famous writers would come, and there'd be an evening where they would read from something they've just published, and there would be an interview. And then the audience could ask questions. And Simon Armitage was there once, and I remember him saying that a poem is something that you put out there into no man's land, and somebody else is on the other side of no man's land, and they take the poem, but there's this nebulous area in the middle where things can be interpreted in whatever way, in either direction. I thought that was an interesting way of looking at it. Also, as somebody said once, a poem is a postcard to yourself. And I find that's really, because I started a diary when I started my Nesta Dreamtime Fellowship, because people kept saying, what's your book going to be about? When's it going to be published? And I got very stressed. And then I thought, well, even if I don't publish anything, which is quite likely that I wouldn't publish anything, if I keep a diary, this is going to be a hell of an experience, whatever happens. So I started writing a diary then, and it's got snippets of poems by other people that I read, things I wrote, going to workshops, news cuttings, all sorts of things. And I can now go back to 2004 onwards. And by looking at my diaries and looking at the poetry I was writing, it's kind of like going back to an early version of me that I wouldn't want to do otherwise. So it's really interesting.
Yes, I'm a firm supporter of keeping a diary. I think
Yeah.
and I mean, I don't know about you, probably slightly perhaps better at it in the first place anyway, but um I used to find it quite hard to translate thoughts onto the page. And after about 6 months of keeping a diary, it became so much more fluent. You could just
Yeah.
write your day
And
very easily.
it's a good place to go to reflect as well, because in the beginning, when I was having my meetings with Clive Scott, who was lovely, but I was used to the scientific way of doing something, where if one of my students came to see me with a question, a problem, we'd sit down, discuss it, we'd work out what to do. We might have a timeline, and it's all very businesslike, and then we'd go our separate ways knowing what was happening. And I would go to see Clive, and he would seem to be talking about entirely irrelevant things for quite some time. And then he'd
Like
say,
what?
"Oh, I don't know." I remember him saying once, "Have you listened to Dylan? There's a CD, Dylan Thomas reading his own poetry. Have you listened to that?" And I thought, well, no. And when I occasionally showed him a poem, he generally didn't really say much about it. And then I realized after quite a few meetings, if he said, "You must be very pleased," that was off the scale. But I would come out of these meetings, given that that was my only contact point for quite a bit of the early part of the year, and go home and think, I have no idea what to do. And then I get my diary out, and I had one of those pens with 4 different colours of ink: green for reflection, blue for general maintenance, red for urgent or highly, significant events. And then I tried not to use black because I don't like black. I'm a bit superstitious. So I tried to remember what had been said, or what the key things were that made me feel not right, or what was bothering me. And by the end of it, I generally had a way forward, which might be going to buy Dylan Thomas's CD and doing something else. So I still do that with anything. If you work on the basis that you're trying your best to do things, but you feel something's not right, you're not happy about something, then go and sit down and try and work out why. Because if you're a good person trying to do your best, then what's the basis of why you're feeling the way you're feeling?
Yes, good advice. And just quickly, what does black represent?
Well, of course, black is just a somber color. And I have got this thing about I don't like using black pens. So I end up with these 4 color pens, and then the only color left when the other ones run out is black, and then I have to throw the pen away. Yeah, that's interesting, actually. I need to go back through my diaries to work out where I've used black. Sounds like an Amy Winehouse.
Indeed. Back to black. Yeah. Well, we'll maybe watch that space. So your book, you have written, well, actually, it's a prize-winning poetry collection, Mock Orange, published in 2020. But there was another award you've won which wasn't for poetry. That was last year, wasn't it? The um Novozymes 2023 award.
Yeah.
That was for your scientific contributions. Yeah.
Yes. Yeah.
Um, and then a separate prize that you won for your poetry collections.
Yes, so that I was really pleased with that. It was one of those things where one of my writer friends in Wales just sent me an email saying, have you seen this competition? It's called the Sentinel Poetry Book Competition. Because she knew that I'd been trying to get a collection published for a while. She said, you really should enter it. So I thought, all right, I will then. You know, it was a day before the deadline. And I didn't think anything of it. And I think we had to send in something like 20 pages initially. And then I got an email saying I was one of 5 shortlisted, which meant I needed to submit a full collection in a fairly short space of time.
Mm-hmm.
And I knew that the top 3 would be published. So it was very stressful, but luckily I was 3rd.
Brilliant.
So I got published, and that was wonderful. My publisher has very sadly died earlier this year, I think. But it was very nice to get the publication out. One of the other poets, Phil Vernon, again, so now I'm in touch with him a lot and we exchange our writing. And then I have several other pieces of work that I'm now trying to get published, as you do.
Brilliant. And it's all poetry or into storytelling?
At the minute, it's all poetry, but I'm toying with trying to write prose now, because because poetry does train you to be, there's no wasted words in poetry, which is quite interesting when you then go back to writing scientific papers. It's really helped me do that. But now I'm thinking, what would happen if I tried to write prose? I did go on a memoir, online memoirs course with Lucy Hughes Hallett, which was very good. And I used my diaries I mined my diaries for the various exercises we had to do. So I've learned that I can do it.
Mm-hmm.
But I also want to write about this tree that grows in Chile.
Yes.
So, and I started studying the Chilean Nobel Prize winning poets. So Neruda is one of them. But at the minute, I'm not putting too much pressure on myself, because I think I just need to let it all evolve.
Yes, that sounds quite right. Naturally. So I could probably ask 1,000 more questions, but I'm not sure they do you justice anyway. So let's think. Apart from the poetry, but let's say scientifically, what's next on the agenda?
So having worked on plants and diseases and then moved into understanding how plants make chemistry, the next big push for me is understanding how those chemicals can be used as drugs and how they can be optimized for drug use. Mm-hmm. Because the plant kingdom is a treasure trove of drugs waiting to be discovered. And in these days of genomics and machine learning, coupled with other advances, we are now, I think for the first time ever, positioned to systematically go through the instruction manual of the plant kingdom and turn it into the next generation of therapeutics. So very luckily, I got a £4.5 million Wellcome grant last year, mm-hmm. Which has positioned me really well to move my lab more in that direction. And then I also have this company that I've set up with others, Hothouse Therapeutics. So I think understanding more about the medical world and the impact of plant chemicals on the human body and what that means is, so rather ironically, having said I could never have done medicine, I'm sort of moving in that direction.
Ever so gradually, but yeah. You still won't have to cut up organs.
No, that wouldn't go well. I've written about dissecting a dogfish just to get it out of my system.
Good. And do you have any hopes for the future of science as a whole, or is that coupled with what your next steps are, or are there other ideas that you hope other people might be able to develop alongside?
Well, I think from my own perspective, the Wellcome grant that I got has enabled me to recruit a new cohort of really talented postdoctoral researchers who I'm supporting in, as others in my group, also supporting them in hopefully developing into the group leaders of the future. scientifically, I think it's a very exciting time. We need to think big. We need to grasp the opportunities around us. And I think the days of one scientist having their own laboratory and doing their own little thing, then it's beginning to, people have to be experts in their particular areas. That's very, very important. It's like I was talking about the disciplines before, but we also need to be able to embrace big data and automation and making sense of all these complex patterns out there in the world that we can't do with the human brain. So that, I think that's a really exciting opportunity, but it needs to be, I think it will involve scientists working in different ways, more as large teams, and they need to be properly resourced. And also the interface with industry is important, because you don't want to be reinventing what's already been discovered behind closed doors. So I think it's all very exciting.
It sounds it, and that's a nice note to end on. Professor Anne Osborn, thank you so much for joining us today and for giving us an insight into your world.
Thank you.
Did you enjoy that episode of Lab Matters? If you did, please do send it to the people that you think will enjoy it the most.